Hello again, listeners. Welcome to the podcast of the InnCoCells project. We are still in Nancy. We're still at the 7th project meeting. And for this edition, I'm joined by Christophe Carola, who works at Merck. And Merck is actually the only large industry partner in the InnCoCells project, which brings a very interesting spin on the topic. And I'm particularly interested in talking to you today, Christophe, about what you do at Merck and what the role is in the InnCoCells project, especially being the only large industry partner. So it would be absolutely great if you could briefly introduce yourself, tell us about the department in Merck where you work, maybe talk about what you do in the InnCoCells project and how it affects the rest of the partners.
Okay, thank you, Richard. So my name is Christophe Carola. I've been working at Merck over the last 25 years practically. I'm a chemist. I studied chemistry in France. I was in a chemistry school. Then I did a PhD in bioorganic chemistry in Dublin in Ireland. And finally, I did a postdoc in Max Planck Institute in Göttingen. It was more biochemistry. So I started directly after my postdoc. I went to Merck, and I had this opportunity to work directly in cosmetic actives.
Was this something you were always interested in, the cosmetics field?
It was either pharma or cosmetics. Yeah. Both were interesting for me, but I had an opportunity in cosmetics and in research and development, so which was for me really great. And for me, it was really the chance to continue to work in chemistry and at the same time in biology. So that's what I find great in the cosmetic area is you have these two scientific areas and you can continuously work like this. And what I always found very interesting in cosmetics is that you have the possibility, because there is R, research, D, development, to develop some project from beginning till the end. That's projects which doesn't last more than 2 or 3 years. So like this, you really have the chance to see really the whole development of your substance, of your extract.
Almost sounds like the InnCoCells project is like a hand fitting a glove in a way. Because just to remind our listeners, so the InnCoCells project, obviously, as you know, is about cosmetics, but it is, just as Christophe has explained, it's from the ground up developing new cosmetic ingredients. So it sounds like we're the perfect partners in this endeavour. So maybe actually that's a segue into maybe you telling us a bit about what Merck does in the project, because it's a pretty specialised role, a unique role. And I think our listeners would be quite interested to hear exactly what that is.
Exactly. So we have several aspects several tasks. So I'm currently working in work package 5, which is to identify the candidates which could be interesting for the development of future actives. So our role at Merck first is to organize the in vitro screening.
So for a non-specialized listener, let's say, what does in vitro screening mean, as in a broad definition?
So in vitro screening, it means that you will test the different extract, because we work with a lot of partners. We are 17 in the InnCoCells project, and many partners send us some extracts.
So they have their plant cells, their whole plants, their heavy roots.
Exactly.
And they make extracts and they all get basically posted to you.
Exactly.
And you then...
So we receive different extracts from the different partners, and what we do is we are going to test the different extracts in cell assays. So we are using cDNA microarrays.
That's very technical. What is a cDNA microarray? It's like having all the genes that you want to see if they're switched on or off, or how strongly they're expressed. You can basically apply a cell extract and you can make a microarray rows, a grid of different gene sequences, and you can make it light up different brightness depending on how strong your gene is working. I think is probably a good layman's way of describing it. Would you agree?
Yes, we could do it like this. And so that's at the end, you try to identify which are the extracts which have the most interesting activity on several, on different proteins. So you have proteins which are related to inflammation, and you have proteins which are related to aging. And then you try to see which are we got 90 extracts at the end, among these 90 extracts, which are the top extracts, so the top 15, the top 20 extracts, having a very interesting biological activities on the different markers.
I see. So if I understand this correctly, you use this array and you have your different extracts, 90 different extracts from the other partners, and you're looking for particular genes that are switched on, which are normally switched on if you have an effect which is anti-aging or anti-inflammatory. So you're looking for like a pattern which is indicative of an ingredient which is active in the way you want it to be.
Yes, you see the evolution, you see the way the substance or the extract is stimulating the expression, the gene expression.
Yeah.
So you can have sometimes upregulation in some cases or downregulation in other cases. It depends on the different protein you're looking at.
Is this very strongly correlated with how the product would work on a human? Is there a good relationship in there?
There is a good relationship. Of course, it's an indication. It needs to be confirmed later in other skin models, for example. Here, we are just talking about cells. So it's an indication.
So it's literally cells growing in a dish, and you apply the extract, and you see what happens.
Yes, you have little wells, and you have your different cells. So we were working with the Test Institute and on different cell lines, so keratinocytes and fibroblasts and even monocytes.
So these would be this kind of cells that you would normally find in the skin, and you're seeing what happens to them. I see.
Exactly. Yeah. And after, you can indeed confirm the result you got in 3D model or in ex vivo. So a 3D model, it's a model that you build, that you cultivate under some very precise conditions with a medium which is perfectly defined. At the ex vivo, it's more a biopsy.
So in one case, you're actually making something that behaves like skin from cells. So you're taking different cell types and putting them into a structure.
Exactly.
And the other one,
3D.
in 3D, yeah. Yes. And the other one is actual skin, punched out circle of skin, if you like.
Yes,
and
exactly.
Do they correlate well, the results you'd get in each of those systems?
Well, normally, when we test an extract on a 3D skin model, we never check a second time in an ex vivo model later on, and vice versa. Sometimes we test it in ex vivo and we won't test it in a 3D model. The point is that there are some advantages and drawbacks for each model and each system. So, of course, the 3D system, it's more robust as the ex vivo, because the ex vivo, it's an explant which is taken from plastic surgery, for example.
Yeah.
And of course, depend on the donor.
Do you get very different behavior, for instance, if the donor is like an 80-year-old man or from an adolescent?
Yes, definitely, yes.
And how does that affect the outcome of the test, for instance?
Well, you get different results at the end, and sometimes you won't have. In particular, if you play with the age, you mentioned the age. So a biopsy of someone who is 25 and a biopsy of someone who is 80, you will have, of course, for certain protein, you will have different stimulation, and with the influence of your extract, you will sometimes stimulate more the cells in one of the explant as in the other one.
So now I'm going to sound totally naive, but as a theoretical example, would you ever have a situation where you have a biopsy or an explant or whatever it's called, and you have one, for example, from an 80-year-old man and one from a teenager, and you're looking for testing a cosmetic ingredient that's meant to have some anti-aging effects? So it seems logical to me. Would you compare the results from the two ages of explant and see if you make the older one behave more like the younger one? Is that how it works, or is it basically a case of you only test it once because it's too expensive? Or I've no idea. How does it work?
Well, usually what we do is that we select some explant we know exactly the age of the donor. We do not compare normally, because normally when we use an extract, for example, when we test a cosmetic product, we want to confirm certain activities. It's true that we would tend maybe to work with explant when we are talking about anti-aging, so explant of people around 60, 65 maybe. And then that's what is great with explant or 3D skin model. You have the possibility to immunostain certain proteins. So in order to visualize the protein which have been stimulated. Before we were mentioning the in vitro screening, it's at the gene level, but in the 3D screen model or in explants or in biopsies, you have the possibility to confirm at the protein level what you could observe in the in vitro screening previously.
I think I understand. So for the benefit of our listeners, so genes, they get switched on, they make RNA, and then that RNA makes protein. And it's not always a direct correlation between one and the next. So just because a gene's very strongly expressed and you get lots of RNA, which will be detected by your microarray, you might have no protein for some reason.
Exactly.
But in these models, you can detect the protein basically exactly as it is, the amount that's there, which is the true marker of what's happening in the skin.
Exactly. So it's true, and it's great that you mentioned that. Sometimes you can have some strong upregulation of a certain gene expression of a certain protein, and after you check it in a 3D model or in explant, and you don't see this upregulation at the protein at the protein level. Sorry.
So it's interesting. I can understand the benefits of these human-like systems. So you've got the 3D model, you've got the explants. Obviously, this saves you having to actually test real people. Presumably, you must eventually have to test real people.
Exactly.
Is there some kind of like cut-off of performance criteria before you go from these 3D models or the biopsies into actual living humans? And what is that level that you have to attain with your models before you get the real people tested?
So once you get good results and when you identify interesting extract, having upstimulating some interesting proteins, and when you later can confirm that in 3D model or in explant that this protein could also be upregulated, then you may have a candidate that you would like to develop until the point that it is a product on the market. Okay, so the next step would be, and that's the first point, which is very, very important, is the toxicological evaluation. So you will evaluate, you will investigate the toxicology of your extract in details. So there are certain tests which are standards. Everyone has different companies have different habits. We are at Merck very strict with the toxicology, and so we are performing 5 tests in order to investigate the toxicological profile.
And this means basically some way of qualifying that the product isn't going to harm people.
Exactly. It's the innocuity. So that's the purpose, is that you need to have 5 results. So we test the different substances in skin irritation. So we have a test to evaluate the skin irritation, eye irritation, Ames test, and to test the genotoxicity of the extract. We have also skin sensitization test, of course, and finally phototoxicity is also.
Which means what happens when you're in the sunlight, or?
Exactly.
Right.
It's when you have substances which absorb in UV, and it's important that when you apply them on your skin, that they don't break off in order to provide toxicology.
Yeah. I think right. So basically, the substance itself might not be dangerous, but if it's exposed to sunlight, it might produce something during its breakdown, which is dangerous. I see. Yeah. Mm-hmm. So these 5 tests have to be done, and once you've got those toxicological data, then you know the product is safe. And you can then test it.
Exactly. So the next step is that once we went through these 5 tests and all the results are fine, so it means we can say that the product is innocuous, then you can start to prepare the formulations. So you need, before preparing an in vivo study, you definitely need formulation. So you need to formulate your product, your extract or your single substance in a formulation in oil and in water, or water in oil formulation.
So what you're saying there, if I understand correctly, is that before you test it, you've got to mix the substance with like the background, whatever they're called, excipients that you need, like to make it into a cream or a paste or something.
Yes. So normally you prepare 2 creams, of course. Well, if you want to organize in vivo study, you have a cream which is the placebo, and another one containing the active or containing the extract, which is the verum.
And would you test them both on the same person, like one on the left hand, one on the right hand, or separately?
Yes, for example, or you have also, it's called hemiphase. So you apply the placebo on one part of your face, and on the other part of your face, you apply the verum.
So, and do you tell them?
No.
So it's like a proper drugs trial.
It's a double blind.
They never know what's on their face.
They don't know.
Right,
So
okay.
we say double blind. It means that the volunteers who are going to apply the formulation don't know which is the placebo and which is the verum. And the person organizing the in vivo study also do not know what is the verum and what is the placebo. So that's why it's called double blind.
Presumably somebody must know, because otherwise you won't be able to.
Yes, we know.
So it's use a code word.
We will use a code. Yes,
okay, right. So you put codes on the bottles, but who applies it doesn't know. The person who's receiving it doesn't know. But exactly. In the end, you know what you can still get the readout, and you can tell if there's an effect. That's very good.
Exactly. Yeah. So, and that's the way it works. So at the end, we always working with a test institute in order to organize this test for us. So the process is of course, you have to define the setup. You have to recruit some volunteers. It's usually between 20 and 30 people. Usually an in vivo study lasts between 1 and 2 months, depending on what you want to analyze. So we were talking about anti-aging beforehand. Yeah. So, for example, just for you to see that when you recruit the different volunteers, you recruit them based on their age, the gender, the type, for example, Caucasian, the phenotype also. So there are some people who have a lighter skin complexion as others. And once everything has been defined, then they will apply the cream, both, so placebo and verum, 1 or 2 times per day. And so certain parameter will be analyzed.
That was going to be my next thing. Because we've talked earlier with the in vitro tests, you can take the cells that you've exposed to the active ingredient and you can basically crush them. Mm-hmm. And the cells die, but you extract the contents and then you can see what genes are switched on. And with the skin models, it doesn't really matter if you dispose of them at the end, but you can't do this with people. How do you test the markers? Is it the same markers, and how do you test them?
It depends. Of course, when you are working in vivo, so you have nowadays much more devices which are able to analyze certain parameters, like, for example, the moisturizing effect of the formulation you are using. You have also markers which are going to analyze the biomechanical properties of the skin, so the firmness or the elasticity. You have also, we are talking about anti-aging, of course, you will analyze the effect of your formulation on the wrinkles, if it reduces the relief of the wrinkles.
There's a standardized way to measure this, is there?
Yes.
Okay.
Exactly.
Because I was wondering if it's like, I feel less, is it all subjective? So you literally measure the depth of skin folds and things
exactly.
like this?
Exactly. Yeah. Exactly. And what is also very interesting is sometimes there are also the possibility to measure the redensifying effect of the formulation you're using. So in the dermis, you have collagen. And so if you could observe in the in vitro screening that the extract was able to upregulate the gene of collagen, then if you could see that in the 3D model that indeed you could see indeed that the protein was also upregulated.
There's more collagen.
More collagen. Yeah. And then you would test it. You test, there is a certain device able to measure the density, the redensifying effect of the substance. And of course, you have also pictures at the end. I could see over the last 20 years how the progress with the device, all the possibilities we have and we didn't have for 20 years. And finally, what is also very important, as you can imagine, with all the volunteers, is there is a subjective evaluation. So the people, the volunteers, the person who tested the different product are going to fulfill, to answer to a questionnaire. And so they will say how if the formulation was agreeable to apply, if how they feel, how is it hurts the skin, how they feel, could they see a progress, could they see really advantageous to use one cream over another, etc.
If you've got all the actual data of measuring wrinkles and things, why is it necessary? Because I can imagine a situation where you take 20 people into a room and you say, we're going to let you sit in this soft chair and we're going to cover your face in cream and you can relax for the next hour and here's a cup of tea. So I can imagine that most people after that experience would be feeling great. So it's asking them if this makes you feel great, this product, then it becomes, as you say, subjective. But is it just because of the experience rather than the product? But if you've already got the data about the markers and the depth of skin folds, etc., why do you need the subjective data as well? Was it just a tradition? Or?
Well, I think it's always complementary to any in vivo study, and it's important to see if what we can measure is correlates to what the volunteers are seeing or perceiving. You know, and so that's why it's important to have both.
Very interesting and quite surprising to me, because I assumed that if you're going to give somebody something and they're applying cream to someone's skin, it's going to feel nice, so that's going to be a positive experience for them. So you do see a correlation between the actual empirical data and the
not in every study, but mainly in the majority of the studies there is some correlation.
That's very interesting.
After, of course, you have some other aspects which have nothing to do with the measurement, with the parameter we analyze. It's, for example, if you have a formulation which is, for example, I say like this sticky, it's not agreeable to apply, for example. So all this touch aspect and the color of the formulation, the smell, etc. That's also very important parameters also for the customer later. Also,
it doesn't really matter what the active substance does. If it's unpleasant to use, then people are not going to want to use it. I understand. Yeah, I've sort of gone down this into great detail, down the rabbit hole, you might say, into the all these assays. I'm going to drag us back a little bit into the specifics of the InnCoCells project, because this is the podcast of the InnCoCells project.
Yes, sure.
And so I wanted to ask you, Merck being our only big industry partner, have you worked with the partners in this consortium before, or are you often working in Horizon projects? What's your general impression of, if so?
We were having a cooperation with PAT in the past.
PAT is Plant Advanced Technology. It's one of our industrial SME partners. That's for the listeners who don't know that.
And but I didn't know the other institute, the other research institute, and that's why I find great to have the chance to.
I've certainly felt that we've all gotten very well in the meetings.
Yes.
But what I've really noticed, particularly at this meeting, because for the benefit of our listeners, Merck, the role in this project, I think it's fair to say, is fairly late on in the development pathway. We need to have things to test. That's what I'm saying.
Yes.
So some of the partners work with selecting plant material. Other partners work in making it grow and produce things which could be useful. Another set of partners work to scale up that process. And another set of partners then work to extract things. And Merck comes more or less at the end of that in testing.
Exactly. Yes.
So, and this is the first meeting, I think it's fair to say, where your role has been pivotal, because we are now 3 years into the project. We're now at the stage of having some candidates to talk about and to test.
Definitely, yes.
And it feels like it's been quite a success. So, InnCoCells, obviously, big project, 17 partners. Merck is the big industrial partner, but we have other small, medium enterprises who are developing cosmetic ingredients. So there's a strange bedfellows, and a big company, lots of little companies, all doing their own things, and academic partners. But Merck being in this project, there must be some advantages for you as a company of all this sort of mixture of partners. What do you say?
Well, I find it's great that Merck had the opportunity to work in a European project. For the member of Merck taking part of this project, I found that very interesting, everything we could learn and from the different partners, and I'm sure they learned from us as well.
Yeah, I think that's definitely true. Yeah.
It's definitely, it's the two different culture, and the industry culture and the university academia culture, and I think it's in terms of creativity and to exchange on this very exciting topic, which is sustainable extract. I think for me, I learned a lot also, definitely. That's certainly the partner learned from us as well.
Yeah. I'm really hopeful this kind of like model of industry academic collaboration can be taken forward into the future, because I think it's fair to say, maybe you agree, maybe you disagree, but I don't think any of the academic partners could have taken their work any further than finding things in the lab without your participation. And Merck may have found it more difficult to find the things in the first place to develop if it wasn't for the academics all sort of like playing with things and getting it so far. Certainly in my experience, academic research comes to a kind of like a barrier where it becomes too expensive. and too much risk involved, where a big company like Merck can then really help out with that stage because you've got the assays, which are, I'd say the most resource intensive part of the project. But you get to select the things which you think are most promising by screening. So you have an
overview.
An overview, yeah.
We have an overview of the different extracts, and it's true that we know exactly, we know the cosmetic market also. So we know the need of the customers and what they really want and what are the requirements they are, and we are in contact with different customers, of course. The cosmetic market is a market which is evolving quickly, and there are some trends certain years and other trends other years. At the moment, for example, it's true that a topic like sustainability, a topic like traceability, a topic like GMO-free, so gene-modified organism-free, that's topic which are in every mouth in the cosmetic world. And it's true that all these regulatory aspects is key, and we have the chance to have knowledgeable people able to support us at Merck for this.
When talking to the other partners, the industry experience and sort of like way to handle the regulations, knowledge of the regulations, knowledge of the market, it's always been an important thing from their point of view as well, this sort of collaboration. So yeah, absolutely fantastic. It's good. Christophe, it's been absolutely fascinating talking to you.
Thank you very much.
This is the first time we've done the podcast and had this large industry perspective. So it's going to be quite a different set of topics to what I've talked to other people about. So I'm very grateful that you found the time to come and sit here in the recording studio in Nancy. And thanks very much. And,
thank you, Richard.
My pleasure. And for our listeners, this has been the podcast of the InnCoCells project. Thank you very much for listening, and we'll see you again next time.